Welcome. In this session, I'll be continuing discussing about transgenic animals and how these transgenic animals are helpful in medicine. And in the last session, I have given some side headings, right? The significance of these transgenic animals in medicine, like they are used in studying the normal physiology and development. They are even used to study the diseases, how the diseases are caused, and if it is the case, then we will be able to find a solution or a treatment for those diseases. And then in producing biological products and in testing the vaccine as well as chemical safety. So in this session, I'll discuss each of these heading in detail. So the first one, let's see, to study normal physiology and development. So in this case, these transgenic animals are actually designed to allow the study of how genes are regulated. Okay, so these transgenic animals are designed to study how genes are regulated, right? So I think in the chapter molecular basis of inheritance, we have studied regulation of gene expression. So the gene expression is nothing but the DNA forming mRNA, and that mRNA undergoing translation and then producing some kind of a polypeptide that shows its effect. Okay, so now how that, so when that protein is required, when it is not required, and all that comes under this gene regulation. And of course, okay, the complexity in human beings is much different when compared with the lower group of organisms. So hence we need to study these genes, how they are regulated, especially in case of mammals. And first we try it by using mice. So whatever that here I have given you the side headings, we will be transferring the genes from other organisms into the mice, okay, and then see that how the genes are working in them first, and then basing on that we will be developing solutions, right, or treatments for diseases. And so now uh, we are using those transgenic mice to study normal physiology and development. And here they are designed, so it means that the genes that are responsible for the physiology or the development, that they are being introduced into these transgenic mice and see that how they are working, right? So those transgenic animals are designed to study how genes are regulated. So how genes are regulated, and also how they are going to affect the normal functions of the body, and also to study how they affect the normal functions of the body, right? So by studying these things, we will come to know that, okay, so whether that gene, so how far the gene is successful in the other animals. So in some cases it may not be, so they need to keep on doing a lot of research and then see that whether they are perfectly suitable, okay, for transferring the genes into other organisms, and see uh, how that gene is getting regulated in the organism into which that gene has been introduced, right? Now for example, in this case the normal physiology and development, so we can study how this insulin like growth factor, insulin like growth factor. So this insulin like growth factor, the genes that are responsible for producing this insulin like growth factor are introduced into the mice, and now they are studying, okay, so genes for this factor are introduced. So they have introduced into the mice, and now they are studying that how that gene which is producing insulin like growth factor, and how that insulin like growth factor showing its effect on the body of mice, right? And this insulin like growth factor, so as the name says, the growth factor is very important for the growth. And nowadays many people who are short, so they wanted to become taller. So if this becomes successful, the experiments and whatnot, we can develop a solution for those people who are short or dwarf to become normal, right? Of course, still this whole process is in its research, it's going on. So maybe in coming years they may find a solution, and as they are doing experiments with these genes, right? So insulin like growth factor, so which are introduced into the mice, and they are studying how that gene is getting regulated and how the product, okay, produced from that gene is affecting its normal body functions, right? So that's about to study the normal physiology and development. The next one we shall see how these transgenic animals are used in studying diseases. Okay, so to study diseases. So transgenic mice or animals are used in studying the diseases. So what scientists are doing is they are transferring the genes that actually produce certain kinds of diseases, okay, into the mice, and they are studying how that gene, okay, is showing its effect in those mice. Okay, so here already we have developed so many types of transgenic animals with various types of genes that cause diseases, right? And to see how, okay, genes contribute, so how genes contribute for the development of disease, for the development of disease, how they are going to cause, okay, diseases. So that's they are going to study here. And we have made, what I call, several models to see, okay, for treating cancer. So we have transgenic animals for cancer, right? So we know that there are genes already in our cells, and if it gets activated by okay, accidentally, then it can cause cancer in that person, right? So how and what factors will cause those genes to get activated, and how they are being regulated, and everything that we can study by introducing these genes which are isolated from the human DNA and then into the mice, and see, okay, how they are working or functioning. Now we made, okay, mice that have cancer genes from human beings. So those if means those animals, the transgenic animals, will be given various types of drugs, medicines, and then even we will study that how those genes are being regulated in those transgenic animals, so that we will find the treatment and solution for these cancers, right? And we also developed, because these are very diseases that's very quite difficult to treat them, so than other diseases. So there are lot many diseases that don't have a permanent treatment. For them, we are developing these transgenic animals and doing some research on them and trying to find out a proper solution, okay, to treat those diseases. And we also have transgenic animals for cystic fibrosis. So remember, we studied this disease in principles of inheritance and variation. So it is a genetic disorder that will be inherited from one generation, or will be passed from one generation to another generation. And how this cystic fibrosis can be treated. So that, and so those patterns of inheritance and whatnot will be used in fact here, by using these transgenic animals. So the genes that are causing this cystic fibrosis have been introduced into the animals, and those animals are now being studied, for finding out a proper solution for this cystic fibrosis, and also for rheumatoid arthritis, and also for Alzheimer's disease. Okay, so all these actually are genetic disorders. So these cystic fibrosis, rheumatoid, so these are genetic disorders. It's an genetic disorder. Okay, Alzheimer's disease is also a genetic disorder. Now, with these genetic disorders, to find out solution and permanent treatment, so there are no treatments for this. Of course, for the cancer, we have treatment if it is found in early stages. A complete treatment is available, but for the cystic fibrosis, rheumatoid arthritis, and Alzheimer's disease, they don't have any permanent treatment. And the rheumatoid arthritis, of course, it's a autoimmune disorder. Okay, and of course the causes are not properly known for this disease, and we believe that there are some genetic as well as environmental factors involved in causing this rheumatoid arthritis. Okay, and for these diseases there is no permanent treatment, so we are trying to find out that permanent treatment by isolating the genes that cause these diseases, and then inserting them into the DNA of other animals, which we call them the transgenic animals, and trying to find out the solution for those diseases. Right. So that's how we are using the transgenic animals in finding out solutions or treatments for various diseases. The next one, in making biological products. So biological products. So what's this biological products here, right? See here, medicines, in fact, which are used to treat the diseases. So especially in case of human beings, they contain some biological products. Okay, and these biological products means the products that are obtained from the living organisms, and to obtain these products, it is actually a costly process. So right, and we need to make in this case transgenic animals. So here, to produce those biological products, so scientists have developed transgenic animals, like for example, right, so transgenic animals for producing something called alpha 1 antitrypsin. So it's okay a substance that is obtained from, of course it is generally produced in the human beings, and what happens is the gene which is producing this alpha 1 antitrypsin, they have isolated from the human being and inserted into an animal. Okay, here mostly I said they are all mice. More than 95% of transgenic animals that we have today are all mice, and then make those mice to produce this alpha 1 antitrypsin. So this is a protein, right, and is used to treat used to treat a disease called emphysema. Used to treat it, okay, treat a disease called emphysema. So I think in the chapter breathing and exchange of gases, I think we have come across this word emphysema, which is a destruction of alveoli due to smoking. And by using this protein alpha 1 antitrypsin, we can treat this emphysema. Actually, there is no treatment for this disease, okay, emphysema, right? And of course we are trying to even make certain kinds of biological products to treat other diseases like this phenylketonuria and cystic fibrosis. So still the research is going on, so we are not yet successful. Of course this one is already available in the market, the alpha 1 antitrypsin, and they are already using to treat the emphysema. Okay, so similar attempts are also made, okay, for this cystic fibrosis, and also for PKU, that is the phenylketonuria. So for these diseases we are trying to find out a solution. And then, okay, in 1997, so in 1997, okay, a transgenic cow was developed. So this transgenic cow named Rosie. Okay, so the transgenic cow, okay, is named Rosie, and it's going to produce human protein enriched milk. So this produces human enriched protein milk. It's going to produce, okay, produces human enriched protein milk, right? So of course in these modern days, okay, giving milk to the young one, okay, they don't have time, and they need to get into the job very quickly, so because of this most mechanized lives, what happens is, right, the mothers were not able to take care of their young ones, for that reason, okay, they will be taking up milk, the milk from the cows, which will be not having these human proteins. And because of that, the young ones who are born will be having nutritional problems, okay, or deficiencies. And now to solve that issue, so what they have done, the genes that code for these proteins, right, have been isolated from the human, and then they are introduced into, okay, a cow, and that cow, it's now transgenic cow, and they named it Rosie, and it's producing milk that contains human protein. So that contains human protein, right? And uh, that human protein is alpha lactalbumin. Okay, so that protein is, right, it's called the alpha lactalbumin, right? And now this cow's milk, so as the mothers used to give cow's milk, now they can give this milk without any problem. The baby will not be developing any deficiency problems, because the cow's milk is having the human protein. So due to absence of these proteins in the cow's milk, the young ones will not be able to get proper nutrition, right? So that's how we have solved the problem, right, by using these transgenic animals, okay. Right, so this human enriched milk, protein milk, is nutritionally, okay, so nutritionally more enriched. So it is more enriched, right? Of course, nutritionally more enriched than cow's milk. Okay, so this is how we are using transgenic animals to produce, okay, uh, different biological products. So in this case we have seen two examples of the biological products. So one is alpha 1 antitrypsin, so which is used to treat diseases like this emphysema, okay, and then solve a problem, right, where most mothers like to feed the young ones with the milk of cow. Okay, now that transgenic cow, the rosy, which is producing the milk that is almost similar to the human milk, right? So that's about in making biological products. So we are using these transgenic animals. The next one we'll see about testing the vaccine safety. So testing the vaccine safety. So we know that once the vaccines are made, they should not be released, directly and used on human beings, right? First, they need to be tested and see whether they are safe to be used on human beings or not. So first, after making these vaccines, we need to test on animals here, right? And of course we will be making the transgenic animals that will be carrying that particular disease for which we are using, which for which we have developed a vaccine. So that disease, genes are already introduced into those animals and made them transgenic, and on them we will be finding out whether the vaccine is safe or not. So now for this process, transgenic mice are used. So we are using these transgenic mice to find out the vaccine safety here. And like for example, the transgenic mice for, okay, safety testing of polio vaccine. Safety testing of polio vaccine. For that, we are using these transgenic mice. Now, the point is that if these transgenic mice are successful, right, we will be replacing the monkeys. Actually, in earlier days, we are using for this polio vaccine safety testing monkeys. Okay, so for this polio vaccine safety testing, monkeys are used. Monkeys are used. Okay, right. And of course, maintaining the monkeys, it requires larger space and a larger food, right? And there are many other complications in maintaining the monkeys here. And this is what we used to do in earlier days, but now what we have done is we generated transgenic mice, and scientists are saying that if these transgenic mice become successful, okay, for testing the vaccines, then these transgenic mice will be used instead of these monkeys, right? So we are planning to use these transgenic mice instead of monkeys, right, in near future, right, for testing the vaccines. Then the last one under this is chemical safety testing. So I'll say about chemical safety testing. Okay, so this procedure of chemical safety testing is same as that, okay, testing the toxicity of drugs. And this chemical safety testing, so you can just simply call it toxicity or safety testing. Okay, so here it's also called as these, with the two names. So test, the toxicity or safety testing, one and the same, right? So chemical safety testing, toxicity or safety testing. Now I told you that whatever that procedure we use here, so is same as that of using, or testing the drugs. So drugs, which means medicines. So before any medicine that is generated, so that medicine need to be tested and see the toxicity. So here, I think if you even take a small paracetamol tablet, and they will tell you the dose, so how much dose need to be taken in one day. Okay, and what happens if that dose is taken than the normal dose. So in case of this paracetamol, it can damage liver, and so for that reason, so they suggest that one should not be given paracetamol more than some 1400 mg in one day. They should not take. So this is how they have come to know, is that they will be using these transgenic animals and will be given those drugs and see that what effects will be going on with them, and then note all those side effects of those drugs. So not only this paracetamol, so if you take any drug and just simply type it in the Google, and you will get just type that side effects of so and so drug, then it will show you the side effects of those drugs. And all these side effects, how we have come to know, is by giving these drugs to the transgenic animals, and we recorded this, whatever that problems they were shown, I mean, by the transgenic animals, right? So now, first, what they are going to do, the scientists, they will make the transgenic animals, right, more sensitive to toxic substances. So scientists are going to make animals more sensitive to toxic substances, more sensitive to toxic substance, right? Here, only when they are made more sensitive to toxic substance, will come to know that how that particular toxic substance is showing its effect. And we will record all those things. So once we make animal, so that's a transgenic animal, more sensitive to toxic substances, then these transgenic animals will be given, uh, with the toxic substances. So after making the animals sensitive to toxic substances, so they are injected with the toxic substances, and then later the effects are studied. So the effects are studied, so how they are going to cause the problem. Okay, so what will be their lethal dose. So lethal doses means at what concentration, if something is taken, then it results in the death of the person or death of the organisms. Okay, in mice, what is the lethal dose, and in human beings, what will be the lethal dose. So that all will be calculated, and then before releasing of this, even like pesticides. So say suppose the pesticides that we use in agriculture to kill pests, so that pest safety will also be determined by using these transgenic animals, like these transgenic mice, right? So first they will be made more sensitive to that toxic substances, and then they will be injected with that toxic substance, and then whatever that effects shown by that transgenic animals will be noted. Right, so that's all about the significance of transgenic animals in medicine. So we have seen that how the transgenic animals are used in studying the normal development, okay, or the physiology of organisms. I mean, the genes that were introduced. So they will be studying that how the genes are working, and then studying the diseases, how the genes are causing diseases, and finding out the solutions for them, and producing, okay, biological products. And then we have also seen how they will be created, the transgenic animals that can be used to test the vaccines and also the chemicals, right? So that's all about those transgenic animals. Now let's look at ethical issues. Now what is these ethical issues, the morals that we need to know? So here, uh, whatever the secret of God, the DNA. So we have come to know that the DNA is the ultimate source for whatever the features the animal is having. And we have gone to an extent to even manipulate the DNA however we like, and whatever the organism we like to develop. So that all can be done because we have come to know, what this DNA is and how to manipulate it and what to do with it. So, and as a result, many people, if this information gets into some people, they can manipulate in such a way to develop, different types of, organisms for their benefit. And if they develop such organisms, it can cause, a lot of environmental issues, right? Because the transgenic animals in some cases may be more potent when compared with non-transgenic animals, and they can compete with once they're released into wild, and it can result in the extinction of the wild varieties, and there will be a lot of even ecological imbalance that can take place if these biotechnological processes are not regulated. So hence, we need to develop some certain rules and regulations to follow and develop these transgenic animals. So it is not like anyone can use this. So there are certain rules and regulations that government have laid down, and then everyone should follow these rules and regulations. So to even generate a transgenic animal, okay, you need to get permission from the government. So how far that work, the research what you are going to do is will be helpful, okay, to human beings, whether it can be done or not, that will be all regulated by the government of so and so countries, right? And for this they have set up something called GAC. So this GAC, that stands for Genetic Engineering Approval Committee. So it stands for Genetic Engineering Approval Committee. Okay, and this will approve any research in the country. So that will be present in different. Of course, each country has its own GAC, and they will regulate. Whether they are going to accept that research. So we need to put a proposal first if you want to do research, and they will be going through that research, and whether it can be done or not, that will be all decided, and they have some rules and regulations to be followed. So that is the GAC, Genetic Engineering Approval Committee. So this will make decisions regarding all that genetically modified, okay, organisms research, and the safety of introducing those organisms in, okay, for or for the public services, all that will be under this GAC, right? Now we will see something called biopiracy. Now what is this biopiracy? So here biopiracy means some countries, so multinational, there are some companies, multinational companies, what they're going to do. So they're going to the countries where the resources are very high, and yet that country people are not using those resources very much. Like if you compare with the USA and India, India has more resources, biological resources, when compared with the USA. So those people would like to come to India and try to explore the natural availability, of these certain kinds of substances in, okay, in India. Like for example, the turmeric, okay, and the properties of neem. See, the point is that they did not discover all these things. Indians already know, the tradition that, to treat certain bacterial diseases. And even now there are many people who would like to put neem leaves, okay, in the water, and they like to take that waters for bathing. And even the turmeric powder that has antibiotic properties, so those also will be, that turmeric powder has been used from very very long back. Now what these people have done is, the US or some other country people have come to India, and they took patents on this neem, turmeric, and many other, substances that Indians were using very long back, and they already know what the properties they are having. And once they take a patent, okay, that cannot be used. So however, the sense, right, so they need, that usage of that particular turmeric or the neem will be regulated by the patent. So that is what happened already in past. So just before that, I'll just tell you the definition of this biopiracy. So biopiracy, it simply refers to the use of bioresources. So this is use of bioresources by multinational companies. Use of bioresources by multinational companies, and right, other organizations, and other organizations, okay, without proper intimation. So they should intimate. So if the people who have come to India and they have come across this turmeric or neem, if they want to take patent or want to do something else, then they should inform to the people of India, and then even they should pay for that. So that is compensating. They wanted to use this turmeric powder and like to take the patent and so and so, right? But they will be not paying. So in that case, that is called the biopiracy. So it's like stealing the knowledge of some other people. So use of bioresources by multinational companies or other organization, right? So without right proper authorization. So there is no, okay, proper authorization of that particular whatever, like turmeric, neem, and many other, Indians, traditional Indians, they know a lot of varieties of substances that they used to obtain from the trees, and they use it for various purposes, right? And also without compensatory payment. So without compensatory payment, so they are not going to even pay for that. So if it is the case, then such a condition, we call it biopiracy. And hope we know that India, as I told you, that it's very much having bioresources. And in India itself, we have around 2 lakh rice varieties in India. So there are 2 lakh rice varieties which are present in India, and even there are around 27 varieties of basmati. So 27 varieties of basmati rice that which were already recorded in India. And we know that the taste and the aroma of this basmati rice is so famous, okay, and are used for making varieties of rice dishes all around India. Now, what happened is uh, in the 1997, so in the year 1997, an American based company, an American based company, okay, so that company, when it has come to India, so what they have done is uh, they have taken some samples of that. Of course, what they have done is they crossed the semi dwarf varieties. So they crossed the semi dwarf varieties with the Indian basmati, right? So in 1997, American based company, what they have done is they crossed Indian basmati with the semi dwarf variety, and whatever that new variety they obtain, of course that is not actually a new variety, already those are existing okay, or in India. And what they have done is, so they told that this is a new variety and they have taken patent on it. So of course most of these later the cases were filed against these patents that were taken by some multinational companies by India. So they have lodged a complaint. Even the patent that have been taken on turmeric, the patent that have been taken on neem, okay, both of them who have antibacterial properties, and Indians know it from thousands of years, right? And of course now all those patents are cancelled, including this patent, right? So when they told Indian basmati crossed with semi dwarf, and they told that something new variety has come, that which already are existing in India, right? And all these patents now they have been cancelled, of course a certain kind of biopiracy can even take place in future also. So hence we need to even control this biopiracy. And for this, okay, Indian Parliament already they have cleared, okay, a second amendment. So they have cleared a second amendment on this Indian patent bills. Okay, so this is a general topic that we see the biopiracy, and even what I say about the ethical issues, the morality of using transgenic animals, as we are not allowed to use however we like, and there are rules and regulations to be followed. And also in case of these patents or this biopiracy, so right. So that's all about these ethical issues and biopiracy. With this, we complete this chapter, biotechnology and its application. So in the next session, we'll start with the organisms and populations.